which is why this chapter lands like a punch. Care once carried seeds. Start with a promise that felt like a miracle in the 196s and 197s, human growth hormone for children who weren't growing. It came from cadavers. Teams collected pituitary glands, pooled them, purified the hormone, and injected hope into small arms. Years later, letters started to land like stones. Young adults who'd received treatment as kids began to stumble, jerk, and slip. The link was brutal and specific. Creutzfeldt-Jakob disease acquired not by food or fate, but by care. Worldwide, roughly 226 cases traced to cadaver-derived growth hormone, with attack rates tied to how rigorously each country's batches had been purified. Parents held boxes of childhood syringe caps and asked if saving inches had cost a lifetime. Meanwhile, in operating rooms, surgeons patched skull defects with dura-mater grafts, tough membrane harvested from human cadavers. It was the right material, the right shape, and for a while it seemed safe. Then clusters appeared years later. About 228 cases worldwide linked to those grafts, many tied to a popular brand processed in ways that didn't fully inactivate prions. A woman who'd finally fixed daily headaches after a repair started veering on sidewalks. Her husband noticed one shoe wearing down on the outer edge. By the time anyone connected lot numbers, the incubation clocks had already run long and quiet. There were smaller, haunting parables. An EEG technician in the 197s reusing depth electrodes. A corneal transplant from a donor who didn't have a diagnosis yet. A handful of suspected transmissions from neurosurgical instruments that had once touched high-risk tissue. In each case, certainty bent under time, incubation stretching a decade or more. Medicine changed protocols, but calendars kept writing outcomes already set. Why here? Because prions are stubborn in ways surgical routines weren't built to fight. They cling to steel like memory. Standard autoclaving, reliable steam and pressure that drop bacteria and scorch viruses, doesn't guarantee prion inactivation. Temperatures that melt plastic won't reliably erase the fold. Some disinfectants cross-link proteins, fixing seeds to surfaces. The fix wasn't better scrubbing, it was different thinking. Picture a sterile processing unit after a near miss. A pathologist calls upstairs, suspected prion case. The OR manager stares at a tray log and a bin of instruments scheduled for tomorrow. She lifts the whole set and drops it into a red-labeled container marked for destruction. Thousands of dollars gone in a clatter. She signs the loss and sleeps that night. Guidelines evolved with scars.
When prion risk is on the table, certain neurosurgeries, posterior eye procedures, any work likely to contact high-titer tissue, hospitals isolate instruments, use single-use where possible, and, if reprocessing is unavoidable, follow prion-specific protocols. Long soaks in strong sodium hypochlorite or 1 to 2N sodium hydroxide, followed by prolonged autoclaving at higher temperatures. Even then, many centers choose destruction over reuse. Color-coded tags, prion-labeled bins, and laminated algorithms live at eye level, so a well-meaning tech doesn't drop a suspect wrongdoer into a standard washer and contaminate the chamber. Timing matters too. Dried organic residue shelters seeds. Teams chant a mantra. Keep it wet, get it down, flush lumens now. Not all news stayed grim. The switch from cadaver-derived growth hormone to recombinant synthetic hormone ended that entire iatrogenic stream. Synthetic dura replaced human-sourced grafts. Vendors began flagging prion inactivation compatibility for devices. Education moved upstream. Residents learned prion pathways the way they learn hand hygiene. RT-QuIC brought speed to diagnosis, letting infection control trigger instrument containment early instead of after a biopsy return days later. But lagging clocks still deliver letters. A patient in his 30s opens mail from a national health service 15 years after a dural repair. We are writing to inform you of a potential exposure. He reads it 3 times, then sets it face down. There isn't a test to certify safety, only surveillance and a card to hand, future surgeons, so they can choose tools and protocols that don't turn one mistake into many. Here's a micro story that shows what learning looks like. A Tuesday at 2 a.m., ophthalmology flags a posterior eye case with an atypical history. The charge nurse slaps a red Prion precaution tag on the cart. The The scrub tech swaps in single-use blades, the reprocessor tapes off a shelf, the supervisor schedules a 134 degrees Celsius extended autoclave run for any reusable that crossed the field. The next morning, the department asks about cost. The supervisor slides a one-page incident sheet across the table, cheaper than a letter to 200 patients. Impact ripples, ORs pause rooms, schedules reshuffle, finance signs off on losses measured in trays, not tools. Most notified patients will never get sick. True exposures often vanish under scrutiny, but anxiety ignores nuance. Hotlines open, nurses repeat careful sentences. What we think happened, what we did, what you should tell providers in the future. The lesson is precise. Prions exploited our best intentions by writing pooled biologics, graphs, and steel. We answered with different materials, rewritten playbooks, and the discipline to throw away what we can't trust. Perfect protocols only protect tomorrow, not yesterday. But tomorrow is still everything for the next patient. What a prion really is. If care once carried seeds, then we have to ask what the seed is. Not a microbe with genes, not a virus with a script. The infectious agent here is shape. Start with the protein we all make, the cellular prion protein, PRP. In healthy neurons, PRP sits on the outer membrane, tethered by a lipid tail, folded mostly into alpha helices. It likely helps with signaling or shield cells under stress. Nothing sinister. But under the wrong conditions, some PRP flips into a beta sheet, rich form that stacks into long amyloid fibrils. That misfolded architecture can recruit the normal protein and press it into the same conformation. A fold becomes a template. The template becomes a lesson that spreads. Stanley Prusner pushed this protein-only idea in the 198s and 199s and caught heat for it. Without DNA or RNA, how could an agent vary, adapt, or persist? Yet experiments kept stripping away alternatives. Treat samples with nucleic acid, destroying enzymes, and infectivity stayed. Denature the protein and infectivity fell. Step by careful step, purer preparations and transmission studies built a case strong enough for a Nobel. The argument wasn't poetry, it was stubborn residue that refused to die unless protein died with it. If you need handles, take 3. Origami. Once a bad crease exists, it stamps the next sheet. Sourdough. A tiny starter inoculates a batch, and tearing the loaf creates more starter. A corrupted save file. Open it, and the corruption propagates into whatever tries to read and rewrite it. Add the special twist of prions, fragmentation. Long fibrils snap, creating fresh ends where growth accelerates. Growth begets shattering. Shattering begets more growth. Skeptics asked about strains. Without a genome, how do you get distinct types with different incubation times and brain targets? The answer lives in conformers, alternative, stable, misfolded structures of peer-peer. Each conformer exposes side chains and grooves in a unique way, imposing its geometry on new recruits. Pass a mixed population through a host, and selection favors the conformer that converts fastest, traffics better, or resists clearance. It looks like evolution without genes, because the selectable information rides in folds. Context pushes too. In vitro, purified PRP can convert with nothing else. In vivo, cofactors tweak the odds. Lipids, polyanions, like RNA fragments, glycosylation patterns on PRP itself, and even metal ions can speed or slow templating. None of that reintroduces a blueprint. It changes the stage, so actors move differently. That helps explain why some brains resist. Chaperones stabilize the healthy state. Certain glycans make docking awkward. Inflammation or aging can tilt the energy landscape so the wrong valley sits a little lower. Zoom into the cell. Misfolded assemblies crowd membranes, disturb trafficking, and drag microglia into a chronic defensive posture. Under the microscope, spongiform change. Tiny vacuoles in the neuropil marks neighborhoods where networks collapsed. Toxicity isn't one note. It's missignaling, membrane injury, synaptic failure, and the slow exhaustion of support cells responding to a problem they can't clear. Meanwhile, labs turn the hypothesis into tools families could feel. Protein misfolding, cyclic amplification, and real-time quaking-induced conversion, PMCA and RT-QuIC, offer excess normal PRP as bait, then agitate samples so any seeds recruit and fragment, amplifying a signal a dye can read. In clinics, RT-QuIC on cerebrospinal fluid became a workhorse for sporadic CJD.
Some centers validated a simple nasal brushing, reporting around 97% sensitivity for SCJD diagnosis. A fluorescent curve rising on a screen isn't magic. It's the fold caught mid-lesson. Here's the rub on timing. The substrate is PRP itself. Lower it and seeds have less to convert. That's why lowering PRNP expression with antisense tools looks promising in models. It's also why late intervention struggles. Once seeds saturate pathways, stabilizers and blockers push against the tide already breaking into tributaries. Species jumps slot back into the same logic. Sequence differences between, say, bovine and human PRP create friction at the templating interface, the species barrier. Most conformers can't dock well across that mismatch. Rare ones can. Give them enough tries and amplification loops, and a fit appears. Passage in the new host then selects conformers that bind human PRP more efficiently. You didn't mutate a genome, you filtered a shape. One more scene, small but heavy. A junior tech sets up RT-QuIC plates, adds recombinant PRP, pipettes cerebrospinal fluid, seals the wells, and starts the shaker. Hours later, a faint uptick appears on the trace. She runs controls again. The curve holds. Downstairs, a family gets an answer they didn't want. Fast enough to change how a hospital handles steel and how a daughter plans a weekend. Tests don't create seeds. They reveal the lesson already spreading. And that returns us to why shape matters more than temperature or slogans. You can't burn a fold out of existence with routines meant for microbes. You have to respect its routes, contact with high-titer tissues, steel it loves, amplification loops that hand it extra tries, and environments that shelter it. When you see prions as a protein that learns and teaches, prevention stops being mystical. It becomes choreography. Starve the teacher, block the classroom, and don't pass the notebook. Crossing the barrier. Species jumps and strains. While labs proved a fold could teach its neighbors, the world kept asking the harder question, how does a shape in one species find traction in another? The answer sits in a tug-of-war between sequence and structure, the species barrier, and in a cast of prion strains that behave like personalities without genomes. Start with the leap nobody could ignore. BSE in cattle became variant CJD in people. Human and bovine PRP differ at a handful of residues. Those tiny changes make docking awkward, slowing, templating. That friction is the barrier, but it isn't a wall. Feed enough seeds through enough chances, industrial loops, repeated exposures, and a rare conformer finds a way to align. In humans, that docking wrote a young clinical picture. Psychiatric changes first, distinct pathology, and a timeline that traced back to late 198's and early 199's dietary exposure. One fit selected from a noisy crowd stepped through. Here's the plain language model. Think two puzzle sets that almost match. Most pieces don't fit, but if one piece wedges in by chance, it helps the next piece align. That first unlikely fit is the crossing. Passage through the new host then acts like audition rounds. Conformers that bind human PRP better propagate faster. Misfits fade. You didn't mutate a genome, you filtered a shape. Scrapie reminds us the barrier matters. Sheep have carried scrapie for centuries without clear human transmission in the modern era. Sequence mismatches and the strain repertoire in scrapie have so far mostly stalled on our barrier. Yet pass scrapie through rodents with different PRP sequences in the lab, and you can watch incubation times shift and pathology split as selection favors conformers that fit each new substrate. It's evolution's rhythm without DNA. Strains force the identity question. Without genes, what makes one Prion type different from another? Architecture. Misfolded PRP can settle into alternative stable conformations, conformers that dictate surface features, stability, fragmentation rates, and cell targeting. Those structural fingerprints impose themselves on new recruits, so a strain's look and tempo can persist across passages. In practice, that shows up as incubation differences, brain region preferences, and distinct protease resistance patterns in the lab. A bench story captures the patience it takes to see this. A tech plotted onset curves for mice inoculated with a panel of isolates. First passage looked chaotic, wide ranges, messy lines. Her mentor said, "Don't trust first dates." On second and third passages, the curves converged. The conformer best matched to that mouse's PRP stepped forward. Noise gave way to a clean slope that defined the strain in that context. Tissue distribution writes policy in the real world. Classic BSE and sporadic CJD concentrate highest titers in central nervous system tissues, brain, spinal cord, dorsal root ganglia, eyes, driving specified risk material rules. Variant CJD complicates the map. Lymphoid tissues like tonsils and spleen can carry seeds early, which gave tonsil biopsy diagnostic value and explained why blood transfusion, rare as it was, proved possible in VCJD. The map isn't fixed across strains. It's a travel itinerary shaped by conformation and host biology. At the molecular interface, small details swing big doors. A single residue swap at a key contact can slow conversion. Glycosylation patterns on PRP can shield or expose surfaces. Membrane positioning via the GPI anchor affects how often normal and misfolded forms collide. Some models invoked an auxiliary factor, once nicknamed protein X, to explain cross-species constraints. Others showed that sequence differences plus conformational selection could do the job. In life, both scaffold and neighborhood chemistry matter. Caution belongs here. In vitro amplification tools, PMCA, RT-QuIC can show that seeds from one species convert human PRP substrate under certain conditions. That's sensitivity, not destiny. A fluorescent curve in a tube lights up possibility. It doesn't guarantee human disease. What it does is tell regulators where not to build amplification loops that give rare fits more tries. Microstory, 20 seconds. In a surveillance lab, a plate lights up when bovine-derived seeds meet human substrate PMCA. The scientist doesn't run to a microphone. She circles 3 words in her notebook. Possible with help. Then she emails the policy team one sentence. Keep SRMs out of food. That's strain talk, meeting public health. Strain behavior also bends clinical storytelling. Variant CJD's pattern in younger patients wasn't random. It reflected a conformer tuned by selection to our PRP and our lymphoid map. Familial mutations shift the substrate beneath the interface, which helps explain why D178N plus methionine at codon 129 steers toward thalamic devastation in fatal familial insomnia, while D178N plus valine drives a different course. Same alphabet, different fold, different itinerary. For families who ask, how did this reach us? The careful answer is, through a rare conformer that overcame a sequence barrier, then refined itself by passage. For farmers and regulators, the takeaway is simpler. Never build systems that hand a rare fit infinite auditions. Close feedbacks, remove high-risk tissues, respect long incubations. Unknown to many city dwellers, out beyond field edges, another Prion has been practicing this math for decades, moving mouth to mouth, hoof to soil, soil to plant, leaving seeds that wait through winter. Its human barrier remains an open file on every wildlife officer's desk. The deer that don't die right. Chronic wasting disease, CWD. Out past the last mailbox, in a stand of aspens, a buck stood broadside and didn't run. His ribs showed. Saliva hung in strings from his jaw. When he finally moved, it was slow and wrong. Legs too long for the ground, he knew. Hunters call it the stare. Wildlife officers call it suspicion. Labs call it chronic wasting disease. CWD isn't new to deer, elk, and moose, but its footprint keeps edging outward on agency maps, county by county, year by year. The stakes land in kitchens that rely on venison each winter, in towns that sell tags and gas and hot coffee to hunters, and in the quiet trust that wild food is clean. Another stake lives in uncertainty. There are still no confirmed human CWD cases. Uncertainty doesn't calm a dinner table. Here's how the shape moves without a slaughterhouse. Infected cervids shed prions in saliva, urine, and feces. Everyday biology turned into a dispersal network. A scrape on a branch, a lick on a mineral block, a puddle in a trail. Seeds slide from mouth to mouth and ground to hoof. Carcasses left on the landscape add a heavier pulse. Scavengers drag tissue. Winter and thaw turn a rib cage into a slow release packet. Soil isn't neutral. CWD prions bind to minerals, clay especially, and that binding can boost infectivity. Plants can pick up seeds on roots and surfaces, turning a food plot into a reservoir. Months later, a healthy doe noses the same patch, and takes a lesson in the wrong fold. A wildlife officer told a story that sets the tone. He followed a GPS caller's death ping to a fence line where a doe lay under Russian olive. When he cut a piece of brainstem and sealed it, the wind carried alfalfa from a nearby field. He burned the tall grass along the fence to clear the area before removing the carcass. One small firewall against a protein that ignores water, winter, and time. Farmed cervids make the map sharper. Close quarters magnify contact. Shared feed and fence line nuzzles multiply chances. One positive test can lock a gate and trigger quarantines. Sometimes whole herds are depopulated. Disposal becomes its own headache. Prions shrug at most landfills and compost piles. High-temperature incineration helps, but access and cost throttle options. Agencies argue over burial depths and liner specs, while communities argue over trucks hauling animals nobody wants near their soil. Meanwhile, hunters face a freezer dilemma. A dad in Wisconsin gutted a heavy buck and noticed enlarged nodes in the head. At the registration station, he handed over the nodes and went home to wait. 2 weeks later, the letter came. Positive. He stood in the garage with the chest freezer humming and did the math on grocery bills, then carried the meat to the landfill's designated pit. He didn't need a lecture on absolute risk. He needed a rule he could live with. Science keeps probing the species barrier because policy and panic hinge on it. In vitro assays, PMCA and RT-QuIC, can show CWD seeds converting human PRP substrate under specific conditions. Transgenic mice engineered with human prion protein mostly resist, but some experimental systems hint at cracks depending on design. Macaque studies have been mixed. Methods differ, timelines stretch. In 2024, an NIH team reported that CWD seeds could infect and propagate in human-derived brain organoids under certain lab conditions. That doesn't equal a human case. It does remove the comfort of impossible. So what do agencies say in that gap? They don't soft pedal. Test where programs exist. Avoid high-risk tissues, brain, spinal cord, eyes, tonsils, lymph nodes. Don't saw through the spine. Don't split the skull with the same blade you'll use for steaks. Wear gloves. Keep designated knives for field dressing. And deboning. If a test comes back positive, don't eat the meat and don't feed it to pets. These aren't theater. They add friction to a pathway prions prefer. Hunters adapted faster than some experts expected. Check stations that once felt like a hassle now run like pop-up rituals. Coolers open, tags scanned, nodes snipped, advice traded. A teenager in blaze orange watched a biologist press forceps into a jaw hinge and asked, "Is it safe?" The answer was exact. "We're managing risk until we know more. Get your deer tested. Avoid the head and spine. Wait for results." The kid nodded like he'd been handed a secret. It wasn't a secret, it was a system. Policy lags and leads at once. States roll out carcass transport rules. Don't haul whole heads across county lines. Leave spinal columns where you harvest. Bans on baiting and feeding reduce nose-to-nose contact. Farmed cervid surveillance tightens. Movement permits stacked paperwork on fence posts. Not every change is popular. Some feel like punishment for problems born miles away. Disease doesn't care about blame. It cares about contact and time. If you want a pocket map, carry this. Test where programs exist. Don't consume meat from a positive animal. Keep brain and spinal cord out of your grinder. Bag and dispose of remains through approved channels. Ask processors how they segregate and clean. Share results with neighbors. None of it guarantees an outcome. All of it lowers the number of lessons a bad fold gets to give. Back in the aspens, the buck with the stare finally stepped and melted into shadow. The hunter lowered the rifle and checked tag dates on his phone. Somewhere between the trees and the lab, between policy and a plate, we're writing the answer to a question we'd rather not ask. How many chances will we give a prion to try us?